I'm really thrilled to be here today. I do have a presentation, but I just want to start by saying that I'm very privileged to be here today, to be invited to speak to all of you. My talk will be all about where we are, where I think we are going. But I just want to start by saying that I have worked in the field of small cell lung cancer research for over a decade now, almost a decade, and I've never been more thrilled about where we are and where we are going. I think we are at a very exciting phase of lung cancer research and treatment in general, but definitely in small cell lung cancer research. So with that, I'll just share my screen. I want this to be really interactive, so I want you to participate. I don't know how these meetings that usually are, but I welcome you to ask me a question at any point because I really want you all to participate in what I'm speaking. So with that, good evening, everyone. And as Montessa mentioned, I am at the Icahn School of Medicine at Mount Sinai in New York. And these are my disclosures. Some of them will be relevant to what I speak today. So my lab is basically a translational lung cancer research lab, which means we study all aspects of lung cancer. We study cellular reprogramming, which is how cells change themselves in response to therapy. We study the cell intrinsic effects of different drugs. We study how these drugs changes the immune system, but with the goal of developing novel therapies. We use several techniques in the lab: cell biology, flow cytometry, mouse modeling, something that we do very much in the lab. So we use various techniques, but our goal remains, how can we personalize medicine for lung cancer patients? So this is our goal, and we also collect longitudinal we also do longitudinal analysis of blood and tissues from patients to develop mouse models. And I'll tell you how important this is as I go through my research a little bit. As I mentioned, my lab has studied small cell lung cancer quite a bit. So I don't think this disease is new to any of you attending this meeting today, but I'll just put this in perspective with other cancer types. Cancer is only 15% of lung cancer diagnosis. But in spite of being only 15% of lung cancer diagnosis, it is one of the most aggressive diseases. It is the sixth most common cause of mortality in the US. And this is a very aggressive disease. And just to think about how we think about small cell in the clinic. So when a patient is diagnosed, they are now treated with first-line chemo and IO. So they get cisplatin-based, platinum etoposide-based chemotherapy along with immunotherapy. Objective response rate in more than 60% of the cases. But this response is very short-lived because we do see relapse, which is very common. And they are put on IO maintenance, mainly with atezo or durvalumab, which are approved drugs. So we see the maintenance regimen happening, and then upon relapse, we don't have too many drugs that work in second line. And if we look at the percentage of patients that actually respond, we see that the survival rate for extensive stage cases are quite limited, and it has remained limited for quite some time now. So 5-year survival rate is seen in less than 1% of the patients for extensive stage disease. What is that we are looking in the lab when we are thinking about small cell lung cancer? We are thinking about effective therapies. We are thinking about biomarkers, and that will lead to personalized therapy for small cell lung cancer. That's the goal, is to have personalized therapy for small cell lung cancer, very similar to what we have for non-small cell lung cancer right now. Next, I'll talk about effective therapies, which is sort of the backbone of personalized therapy for any disease, and definitely for small cell lung cancer. Of all the failed clinical trials for a small cell lung cancer, there have been 2 positive clinical trials. That's for immunotherapy. So immunotherapy has now been approved as a first-line therapy for small cell lung cancer. And this is one of the 2 positive trials. There were 2 trials, IMPALE 133 and the Caspian study. And 2 immunotherapy drugs, durvalumab or atezolizumab, entered the clinic in combination with chemotherapy for small cell lung cancer. Now, if you look at this curve, maybe it doesn't seem so striking to all of you. And it is not very striking because adding immunotherapy to chemotherapy only improved the survival rate by about 2 months. But it was a shift in the needle in the right direction. And for that reason, immunotherapy was approved in small cell lung cancer. Now we have data for long-term responses up to 3 years. And we do see that there is a separation in the curve. What I mean by that is patients who do respond to this frontline treatment respond for a very long period of time. So we are seeing up to 36 months that there is a separation of this curve, and a subset of the patients do respond long-term to the response to chemo-IO first-line treatment. However, if I summarize all the immunotherapy data, and this is the first-line treatment trials with IMpower 133, Caspian, and Keynote 604, and this is the second-line trials with Checkmate 032 and the Keynote 028, we see that only about 10% patients actually respond to immunotherapy, which means we can do better. We need to do better. We need to bring more patients who will respond to immunotherapy for small cell lung cancer. Now, why do most patients do not respond to immunotherapy? There are many stages of the hurdle. It has been an uphill ride when it comes to immunotherapy for small cell lung cancer. And the reasons are there is lower expression of regulators of antigen presentation, or in other terms, these tumors are not very visible to the immune system to begin with. And then we have very low infiltration of the good immune cells or the infiltrating T lymphocytes. And finally, there is incomplete understanding of immune responsive subsets in small cell lung cancer. We do not know who are the patients who will actually respond to these cases. And we need these information in order to make immunotherapy better in small cell lung cancer. Now, our lab has worked in all these 3 areas. So we have worked on epigenetic priming, or using drugs that can make these invisible tumor cells more visible to the immune system. And I'll talk about that in a couple of slides. And so we are trying to upregulate tumor presentation. And secondly, we have used drugs, the DNA damage response drugs, to reactivate the immune system or to cause infiltration of these good immune cells to the tumor. And finally, we have tried to identify biomarkers of immunotherapy resistance in small cell cancer. So we have worked in all these 3 areas in order to make immunotherapy better in small cell. Now, when you look at small cell lung cancer, and I don't want to go too much into the science, but what I really want you all to appreciate is that there are 2 subsets of small cell lung cancer tumors. Some that have high MHC class 1 expression and some that have low MHC class 1 expression. Unfortunately, majority is MHC class 1 low. Now, MHC class 1 actually makes the immune system aware of a tumor, right? So if you have low of this, which means most of your tumors are not even visible to the immune system. And we have seen through our studies that patients who have this MHC high expression actually respond to immunotherapy better. So we want to shift all the MHC class 1 low to MHC class 1 high. And we have done this by using a drug that targets this target called LSD1. So LSD1 is an epigenetic regulator. What does epigenetic regulator mean? If you think about genes that encodes for protein, now there is an extra layer. Epigenetic regulators control your gene expression. And these targets, actually, LSD1, it is an epigenetic regulator that helps the cancer cells to proliferate or to differentiate. And it is seen to be a good target for other diseases. Now, we have used drugs against this epigenetic regulator LSD1 in our mouse models to show that when you use this drug, ORI-1001, either as a single agent or in combination with immunotherapy, you're able to decrease the tumors in mice. And it is very consistent that you see with this combination, the tumors are much lower in volume than that of the ones that are not treated with this drug. which means this drug is actually very promising in reactivating the immune system. Now with the second, and we have done a lot of work over the years on DNA damage response pathways. So your cells cycle and your cells undergo DNA damage. Now there are several genes or proteins that control the repair of your DNA damage. Now in small cell lung cancer, targeting these proteins, which are the DNA damage regulators, is very good in small cell lung cancer because these cancers, they do have a genomic instability. So what we have seen in a previous study that this particular target, CHEK1, and drugs that target the CHEK1 is very good for small cell. And this is just a mouse study that I'm showing you that this decreases tumors in mice. And as a biomarker, MYC can be a biomarker, which means higher MYC expression can predict that these drugs will do better. So CHEK1 inhibitor is a target, and then MYC is a biomarker. This is an entire field that was launched, and these are some of the trials that are either targeting CHEK1, ATR, or V1. These are all DNA damage response regulators. You can go into clinicaltrials.gov and you will see all these trials here that target CHEK1, ATR, V1, and PARP. So there was an entire field that we launched. Now, next, we know that when there is DNA damage, you can cause infiltration of these good T cells or the good immune cells that I was talking about. So our hypothesis was that if you now combine this with immunotherapy, you have a real potential for killing these tumor cells. So we combined PARP inhibitor. PARP is also another DNA damage response gene. So we combined inhibitors to PARP and CHEK1, and we treated it in our preclinical models. Now, what you can appreciate here is that this red line. So when we treated our mouse models with a PARP inhibitor in combination with immunotherapy, we were able to clear tumors in mice. So the tumors actually vanished completely, and they stayed vanished for over 3 months. And this is really, really exciting because for the first time, we saw that we are able to clear tumors in a mouse model in small cell lung cancer. And this, again, led to various studies that we did with PARP inhibitors, with CHEK1 inhibitors, and now with V1 inhibitors. So we know that targeting these DNA damage response proteins is very promising for small cell lung cancer. And it also, when combined with immunotherapy, can make immunotherapy a lot better. Again, there are various clinical trials that was launched based on our study. You can go into clinicaltrials.gov and you can see this list of clinical trials that are available, which combines the DNA damage response inhibitor with that of immunotherapy. So now we have talked a little bit about the effective therapies that are out there. But an effective therapy is not as effective if you don't know the patient population that it would be most effective in. And that comes the question of biomarkers. Which is the patient population where this drug would be more effective? Or even more important is that you don't want to waste time, right? So you want to know who are the patient population that will not respond to this drug. So that is where biomarkers are so important. About non-small cell lung cancer, there are various drugs that are in the clinic. You must have heard about drugs that target EGFR, drugs that target KRAS. And in non-small cell, we are able to do that because we have these mutations or gain of function mutations where this drug can be effective. But when it comes to small cell, it's a completely black box. And I'll tell you in a minute why it is. We are not able to see biomarkers when it comes to small cell with our knowledge of mutation. And why is that? Because, and you may not understand what these figures mean, but what I'll tell you to a scientist what this looks like, it looks like a nightmare because there is a lot of mutation. And these are all loss of function mutations. So in other words, we cannot design drugs that target these genes because they are loss of function. So there are no drugs that can actually target these genes. So small cell is very different from non-small cell. So you may hear about these drugs that are really effective in non-small cell lung cancer. They won't work in small cell because it's not a mutationally driven disease. And that's something I think we all need to understand as researchers, as patients, as caregivers, that small cell is a very different disease than non-small cell lung cancer. Now the field really understands that if you don't have mutation information, we need to still identify biomarkers. We need to still know where the drug would work. So now small cell is divided into 4 major subtypes, right? And these are not based on mutation. These are based on expression. And I'll tell you the hurdles of having an expression-based biomarker. But just for a minute, I want to appreciate how far the field has come. So now small cell can be divided into ACL1-driven subtype, NeuroD1-driven subtype, YAP1-driven subtype, and PAU2F3-driven subtype. Now, when we take biopsies from patients, we actually are able to, and these are all CLIA certified, so we are able to do immunohistochemistry and to say which subtype you would be most effective in. Why do we need to know the subtypes? Because now we have seen that some of the drugs work better in some of the subtypes. So we are trying to get to some of this pie here. So we know that CHEK1, ATR, and PARP may work better in PAU2F3-driven subtype. LSD1, which I spoke about before, may work better in the NeuroD1 or even in the ACL1 subtype. And DLL3, which is a very important target that I'll talk about in a minute, it may work better in the ACL1 subtype. So now we are able to say, depending on this expression-based biomarker, that some drugs may do better if you have higher expression of that biomarker. Now, coming to the drawbacks. When you think about expression, it is not black or white, right? It's not a mutation that it's there or not there. Expression can be high or low, so you need to have a good cutoff, which is often a hurdle in the clinic, is to decide how high is high and how low is low, right? So that is the hurdle when it comes to expression-based biomarker. But we are getting there. That's my point. We are getting there, right? We know more about this disease than we ever knew before. And I spoke about some of these targets. Now, one thing we really need to appreciate, and I have told this again and again in various conferences, is that we need to know more about how small cell becomes resistant to disease. So when you think about a treatment-naive small cell, it kind of looks like this. And now you treat it with chemoimmunotherapy, the tumor regresses, and then it comes back. Now, when it comes back, relapsed small cell is a completely different disease. They become different in various mechanisms. It's a very heterogeneous disease that it becomes different from each other. Small cell like this, like a herd of sheep. And we can do that for treatment-naive cases, right? But when it becomes resistant to therapy, it's a zoo. It looks very different. Each patient is different from each other. So a relapsed small cell has to be treated in a very different way. We need more biomarkers. We need more information about how these patients are becoming resistant. So I think a lot of the research will go into identifying the mechanisms of resistance in small cell. There is a very, very big hurdle in understanding this. Getting tissue from a relapse to this particular phase of the disease is almost unheard of. Clinical trials don't do that. We don't mandate biopsies in a relapse setting because at that point, we just want to go to the next therapy because we want the patient to get better. So we don't do biopsy at this stage. But it's very important to know how the patient has become resistant. So I think the future is blood-based biomarkers that has shown promise. Now, small cell, since it's very aggressive, there are a lot of tumor cells in the blood. So I think now every clinical trial will request a blood draw from the patient once they have become resistant to a disease or even when they are entering a clinical trial. And I really urge you as a researcher to participate in those mechanisms, is to give your blood so that we can then study your disease. We can see how you're becoming resistant to the treatment that you have received. So I think blood-based biomarkers will be the future. We can do a lot of studies with the circulating tumor cells that we see in the blood. There have been several studies that now shows that we can get circulating free DNA from the blood, and we can do methylation profiling, which can tell us a lot about your disease. What are your subtypes of the disease, how you have become resistant, and what could be the next line of therapy. So these are all the information that we can get from blood. In our lab, now we are trying to build an entire patient-derived xenograft program that will collect circulating tumor cells, surgical specimens, pleural fluids, biopsies, autopsies, and neuromets, where we are able to grow these in mice. And we are able to do drug response studies and all kinds of analysis. We are able to look at the genome, the transcriptome. We are able to predict what therapies would be most effective. And we are also able to identify mechanisms of resistance. And if there is metastasis, how does metastasis happen? So we are able to now do that. And my goal is to develop a PDX program where we are able to do all of this. Now, let me summarize. What is the state of small cell? What are the new targets? What are the promising targets? And what you may be looking out for in the future? Immunotherapy combination, combining PD-L1 with CTLA-4 or with TIGIT. I don't think that's the way to go, because we have seen 2 clinical trials, which is the Caspian study and the Skyscraper 2, which has combined either CTLA-4 or TIGIT. None of them have been really effective. So combining TIGIT or combining CTLA-4 may not be the way to go. Genetic modifiers. I spoke about LSD1. There is another one, EZH2. Can these be good targets? Maybe. We need to see the clinical data. But maybe these are good targets when they're combined with immunotherapy. They can rescue the immune system, or in other words, they can make the tumor more visible to the immune system. I worked a lot with DNA damage response. I believe in these targets. CHEK1, PARP, or V1, ATR. Are these good targets? Most likely, they could be good targets for small cell lung cancer. Now, small cell lung cancer has come a long way. I think I painted a very sort of dreary picture, but we have come a very long way. In 2023, there are 226 clinical trials that are open or pending activation in clinicaltrials.gov for small cell lung cancer. 226, that's unheard of. 179 clinical trials are currently recruiting all over the country and in the world. There are 179 clinical trials that are actively recruiting patients. It's a completely new era where we are looking at targeted therapies that are not dependent on pathway activation. We are looking into BiTEs, CAR-Ts. We are looking into antibody drug conjugates. And some of the targets that you will be hearing a lot if you go to these scientific meetings are DLL3, CES6, CCAM4. These are all the targets to look out for. So these can be the bispecific T-cell engager, which are BiTE. So BiTE is bispecific T-cell engager. CAR-T is chimeric T-cell engagers, and then antibody drug conjugates are ADCs. Just to give perspective, these are how many trials are there for small cell that targets the cell surface antigens. With DLL3 targeting alone, there are 9 trials. With TIGIT, there are 5. With fucosyl GSM1, there are 3. With LAC3, there are 3. With TROP2, there are 2. With B7H3, there are 2. And with GD2, there are 2. So there are so many trials that targets just these cell surface antigens. And I think this is an extremely interesting area of research, because as I told you, mutation is not the way to go. But the cell surface antigens that are specific for small cell could be a very good area of targeting this new age targeted therapy for small cell lung cancer. Finally, what are the conclusions and the next steps? Despite the approval of immunotherapy, most small cell patients still need more effective treatments. So we need to push the tail of the curve. That tail that I showed you, it's still touching the axis. So we need to push it up. We need more patients to respond to immunotherapy. That would be our goal. Personalized biomarker-driven clinical trials are a critical need. Unless we can tell which patients will respond to therapy, we are not going to be able to have all patients respond to that therapy because we need to know the subset of patients who will respond. So the molecular subtypes of small cell could be a way to go, which decides, okay, if you are ACL1 or PAU2F3, you may respond to a certain kind of treatments. Novel therapies that I spoke about, BiTEs, CAR-Ts, and ADCs, really exciting area. And blood-based biomarkers, I think, will be the future when it comes to identifying resistance mechanisms for small cell lung cancer. Biology of treatment-resistant small cell, absolutely critically needed. We need to understand why most of these patients do not respond to therapy and in a very short period of time. And I think as biologists, as scientists, that is where we are actually, giving our resources is to understand resistance to targeted therapy, resistance to immunotherapy. We are entering a very exciting phase of small cell research and treatment. And I believe we will see many breakthroughs in the next few years. It is almost a golden age when it comes to small cell lung cancer, understanding the disease, understanding therapies. We have never understood the biology like we do now. So once we understand the biology, treatment cannot be very far behind. So there is hope for small cell lung cancer. And I think we are entering that age where we can truly have personalized therapy for small cell lung cancer. With that, I would like to thank, of course, my lab members. I mean, they're fantastic. They're working tirelessly to understand these mechanisms of resistance, to understand how these therapies become, what therapies would be most effective, and to what treatment population. And all my funding agencies, research is very, very expensive. And small cell lung cancer does not get as much research funding as I think it should. So all my funding agencies, and I think I'm really thankful to all of them. And of course, patients and their families, because without them, our research would not even happen. So I want to thank all patients and their families. And I think Lynn mentioned this, but those of you who are in New York or even surrounding New York, I think, as a group, if we can have a visit to the Sen Lab Day, I'll be very happy to walk you through my lab, to talk more about the research, to make you meet our team. So I really invite all of you to come and visit my lab in New York, and we can do this over email. This is my email. Feel free to reach out to me. You can do it through Lynn also, who is sort of coordinating this group. But I really want you all to engage with researchers like myself and understand what we do, how we do it, and how much we are actually, pushing the needle. We really want to bring therapies to the patients as quickly as possible. So please come, visit our lab, support our research, and see for yourself, talk to the researchers in my team and see for yourself how tirelessly we are working to bring therapies for small cell lung cancer. With that, I would like to stop here and I want to take questions, any questions that you may have.
And I have a question, and there are also some in the chat. So thank you. As I said, I love it when Dr. Sen comes and talks, and it is exciting to see this new news coming out. I have a question. One of those slides you showed about the clinical trials, I saw about, from my understanding, the kinase inhibitors did not work the same in small cell, but one of those looked at FR, EGFR, and some of those other kinase inhibitors. Is that for people that had non-small cell that morphed into small cell?
So there are trials right now. That's a very good question. And I did not have time to speak about it in detail. So now we are seeing that some of the non-small cell lung cancer patients, once they become resistant to drugs like osimertinib, they become small cell lung cancer. They change to small cell lung cancer. So some of those clinical trials will look at EGFR targeted therapy and then for small cell therapy for these transformed small cell cases. You're right.
Okay. Oh, good. And the question in the chat, why isn't biomarker testing available in the UK? In America, is it standard practice for each individual then? No, it's not, but go ahead.
Yeah, well, UK system it's a lot different. And I think these biomarkers that I'm talking about, these subtype biomarkers, they are CLIA certified. We do this, but we don't know them enough to make clinical decisions yet. So are we looking at them in the clinic? Yes. Can we make real good predictions about, based on these subtypes, of what drugs they should receive? I don't think we are there yet. I think we need more data from clinical trials to guide us to make those decisions. So we are not, the subtypes of small cell are not ready for clinical prime time yet. We have a little bit more to go before they can be prime time.
Now, we do advocate for non-small cell lung cancer, though. You probably have heard, I'm not sure if you've heard that advertisement, but for them to have biomarker testing for sure.
Yeah, and I would love to see that because, as I said, personalized therapy cannot happen without biomarker information. So I think we are getting there with trying to identify biomarkers for small cell. That has been a hurdle historically.
Mm-hmm. Any more questions? I can't see everybody's hand now because we have so many people in the meeting, but you could unmute.
I've got a question.
Okay. Go ahead. Okay.
The mice study that you did, were they getting immunotherapy plus the drug where the tumors disappeared?
Yes. So where the tumors disappeared, they received immunotherapy along with a PARP inhibitor, so olaparib, which targets this DNA damage response protein PARP. And yes, so it received it in combination with immunotherapy.
Are there any human clinical trials going on with that drug?
Yes, that was the slide that I showed you next. There are many clinical trials that are going on combining PARP with immunotherapy based on our preclinical study. We don't have full data yet about whether they responded in patients, but there are trials that are going on. Yes.
Okay, one more question. I've had 4 chemo treatments with immunotherapy, and he scheduled me for 2 more, so it'll be a total of 6. And then I guess I'll go just to immunotherapy. If you decide to do a clinical trial, when would be a good time to do it? After the chemo's finished, or what's a good time to start a clinical trial if you decide to?
Yeah, so I think mostly in the maintenance and in the second line, that's when most of the clinical trials would be. But there are many criteria of getting included in clinical trials, and that's something you will have to talk to your oncologist, whether at that point, what clinical trials you do qualify for. But yes, in second line, in the maintenance and beyond, there are many trials that are looking at some of these combinations.
Okay, thank you. That's all I had.
Absolutely. Thank you.
I have bladder cancer, small cell bladder cancer, and I'm assuming that small cell encompasses all, lung cancer and bladder cancer and different types of where it originated from. Am I correct about that? It's treated the same. I'm currently on Keytruda, the immunotherapy drug.
Yeah. So most of them. So when we talk about small cell, we're talking about the neuroendocrine tumors. This is sort of an unifying factor. They are sort of treated in the same way, but you also have to talk to your oncologist to make these extrathoracic, which we call, other small cell. They, based on what markers you show or what mutations you have, it may be tailored to your particular disease. But you're right, small cell, they're all neuroendocrine tumors, so they're often treated in the same way.
Hi, Dr. Sen. I'm wondering, if I wanted to participate in a trial, would I have to travel there, or do you ever partner? For example, I'm close to Yale, and if you needed blood, could I go to Yale and give the blood there and then they send it to you, or what, something like that?
Unfortunately, it should be that simple, right? But unfortunately, it's a little more complicated than that because, since we are dealing with, patient samples, and they're very much, we have to honor the confidentiality and everything. So unless I partner with Yale and there's a long drawn paperwork and they are all in place to protect you, to protect the patients, right? So if the trial is in Yale, then your sample would most likely stay in Yale unless I have an upfront collaboration or an understanding with Yale that I would get your samples, which would more often not be the case. Because if it's a clinical trial that has been sponsored by a pharmaceutical company, then probably they would get your samples. And it depends on what has been built into your protocol. And probably they will get your samples. And then Yale investigators, who are the principal investigators in your trial, would get to see that. But if you do come to Mount Sinai, and if you get your treatment here, then most likely your sample will come to my lab because I do small cell lung cancer in Mount Sinai. So inter-institution, it's a little complicated.
How often would one expect to have to travel to Mount Sinai?
It actually depends on which clinical trial you're enrolled in, what specific drug you're receiving. It depends how you're responding to that drug. So it really depends on which clinical trial you're enrolled in. It may be more intensive. Some trials are more intensive than others, and you may have to stay back. And you would also have to see whether that trial is even open at Mount Sinai or not. So,
and would the treatment be based on the biomarker?
So first line is not biomarker based, right? So everyone will get chemoimmunotherapy. That's a standard of care first line treatment. For second line treatment, there are a very small number of trials. You may or may not qualify. They may or may not be open at all trial centers. And you will be enrolled based on what your disease looks like. And now there are approved drugs in the second line, like topotecan or lurbinectedin in the US. You may get lurbinectedin in the second line treatment if you do not respond to the first line treatment. So that's a standard of care. But to be enrolled in a clinical trial, you have to talk to the doctors, your oncologist at that center where you are going for your first line treatment.
Thank you, Dr. Sen. Thank you, first of all, on behalf of all of us and researchers, amazing, and your passion for the work that you're doing. We so appreciate it because, it's a very aggressive disease and, we don't have, some of us, have limited time. So it's just so great to see your passion and everything that you're working on. My question for you is, you mentioned the PARP inhibitors being an adjunct with immunotherapy and then maybe with a chemo. So like in my husband's case, he's failed a few lines, although he was highly responsive to carbonatopoxide and immunotherapy for a few months and failed, then went to another immunotherapy, which worked for a little while, failed, lurbinectedin, failed, after a really nice response to all of these. So now we're on fourth line, which is concerning. But the question always becomes, if PARP inhibitors and immunotherapy, why is it kind of like standard of care is in such an aggressive disease that we can't do more kind of out of the box thinking around dropping in a PARP or a new immunotherapy to try? Because if someone's a responder to chemo and it kind of brings it down, we need something to keep that lasting effect. And I'm just questioning, where is the research going on, being able to trial different things in clinical setting outside of clinical?
Yeah. That's a very interesting question, and I'm sorry to hear about your husband. So, when a patient first comes into the clinic, the doctors really want to control the disease, and chemotherapy and immunotherapy, the patients do respond in frontline. So you want to get the disease to regress. You want the disease to respond. And I have shown you some of the data that some patients do respond. We have 3-year, 4-year, even 5-year responders on chemoimmunotherapy. So we don't want to lose that chance. So every patient is treated with chemoimmunotherapy. Objective response rate is more than 70%. Some of them have long-term responses, but unfortunately, we see relapses more often than we would like to. Now comes to the question of second line. We don't understand resistant disease as well as we should, right? So that's why, since we don't understand, we don't know what to give them in the second line. PARP inhibitors work, but there are trials where PARP inhibitor has failed. Is it because the patients were not a right candidate? Is it because it was too late? Were they given where the disease was too aggressive and nothing could have stopped it at that point? Could be. These are all great questions that we need to answer. But now we are thinking out of the box, right? So we are putting PARP in the maintenance regimen. We are putting ATR in the second line regimen. These are all DNA damage response inhibitors. We are putting them in the second line regimen in combination with immunotherapy to make these better. But these trials are not open everywhere. That's a practical problem. They're open in only a few centers. There are only a few spots, and we have to find the right candidate for that trial. So all of these are practical problems that makes my head burst when I'm thinking about a researcher that this should be easy. This should be easier than this. But unfortunately, clinical scenario is a lot more difficult because there are resources involved. There is research involved. There is capability of a center. I'm in New York and I'm blessed to be in a center. I have been at Memorial, now I'm at Mount Sinai. They're extremely well-resourced. So I'm, blessed to be in places that have the resources to do these trials. But there are centers who don't have access to these trials. They're just not open. So which means the patients in that catchment area, they don't have access to these trials. And these are all like very, very practical problems. But we are thinking outside of the box. We are thinking about CAR Ts. We are thinking about ADCs, where most patients will respond.
Yeah, we're definitely thinking out of the box. I think in clinical practice, it's very hard to think outside the box because you're bound by so many of those practical concerns that you mentioned. And then my last question for you is, you mentioned about donating blood samples. So in my husband's case, I don't know where this is going to go, but, if we could donate blood samples so you could study his disease, how do we do that? Is there a blood bank registry like they have for different diseases that we could donate blood samples as it morphs?
Yeah. Has your oncologist talked about it? So, blood samples, it's not just you, someone should be able to do something with that blood sample. There should be researcher in that center who has the ability to look into those blood samples. Otherwise, what are you going to do with that blood sample, right? So it is not donating blood, so it has to be a part of your protocol. There has to be resources against in the center. There needs to be sort of a person who does research like myself who are able to do something with that blood sample once they get it. Otherwise, it's a wastage of a very, very, very valuable patient sample. So I think that's a conversation you need to talk to your oncologist that, look, I have listened to talks where they're saying that you can look into the resistance disease from blood. Is that something I should do? Is that something my husband should do? Do you want to study how his disease has become resistant? If we give blood, what will you do with it? How are they going to be useful for research? Do you have such researchers? So talk to your oncologist, talk to where you're being treated as to whether they have even the capability to do anything with the blood. Otherwise, I don't know how useful it will be.
Thank you so much. I appreciate it. That makes a lot of sense. And I definitely will, because I think that would be the best benefit for everybody, as we go through this journey. So thank you.
Yeah. Thank you.
Is there a better time or an appropriate time to seek out a trial? Tomorrow is my 3-year anniversary of being diagnosed. So yeah, that's the way I feel. I've been through the carboplatin etoposide with Tecentriq 3 times. Okay. I had Zepzelca, for almost a year. And I've been getting immunotherapies. I feel like I'm keeping the monster at bay. Is there a better time to go look for a trial? Is it when things don't seem to be working so well with what you're doing? Or is it when you're first diagnosed? Or is there an optimum time for a patient to say, okay, I want to go for a clinical trial?
Yeah. So on biomarker-driven trial, I wouldn't, that's the problem, right? With non-small cell, you have an EGFR, you get a drug. You can ask for that trial. You have a KRAS, you go for that trial. Small cell, you don't have that. So if you are responding to the treatment that you're getting,
Correct.
just count your blessings and, just because unless you have a biomarker, your trial, you asking for a trial will not help you, because unless your disease comes back, we don't know what to do with it because we don't have a direction to go
Right.
unless we
Right.
know that. So I just hope, I come back to this meeting 6 years and you're still like, you're telling me this is my 6th year anniversary. But if you're responding, then I think that's what you should stick to. I don't think that you should be seeking out a trial because it's not a biomarker driven disease. So I don't think anyone would put you on a better trial if you're responding to that disease because they don't have a biomarker to go for.
That would be my concern, is if I'd say, okay, this time I'm going to go for a trial, and I go for a trial and the thing gets ahead of me because they don't know if it's going to be effective. Right. So I'm going to stay pat where I am then.
Yeah.
I want to ask a question.
Thank you.
And I know some of those trials too, depends on what
what you've received before and how you've responded to that before. So there are a lot of inclusion criteria in those trials, and that's a conversation you should having with your doctor.
Right.
Like, I'm responding, what next? And they will probably tell you you're responding. So, we will count our blessings for now.
Okay, I want to ask a question.
Yeah.
Okay, I've been listening to you guys and it's, a little hard because where do I stand? Okay, I am cancer free from small cell lung cancer. They told me that in January. Okay. In the 5 years, I never got sick. After my 7 months, after my 7 months of treatment, which ended in July, from January to July, it was all so happened so fast. I have never been sick. Where do I stand? Do I look for trials? Do I look for that? I try not to look for anything. I'm just grateful. It's very complicated. When I hear people and I'm like, do you understand? Or maybe you don't. I've never been sick. Isn't that weird? So I really don't know a lot of the biomarkers and this, and never thought about trial but then I listened to this and now it's gotten to 5 years. Wow. Yeah. But then again, there's other people that have made it way longer than me.
Right.
But imagine when she said 3 years, what a blessing. I went through those 3 years. When I lived on a prayer, and then one day I said, no, I'm going to live to win. Because they make it seem like it's a death sentence. But it is for some people, I don't know. It's very confusing. Because it is for some people, and I don't really know. I just feel so blessed. And it's so complicated when I hear all these things and all this trial, and I guess I will die from that. But when do you, like she said, 3 years, when does she think of a trial or something? What should I do with my life right now? Think of trials?
First of all, I'm so happy for you that you are disease free.
I'm sorry.
No. I'm just so happy to even see you and meet you and hear your story, because I feel blessed. I'm happy to have known that someone that is actually free. 5 years. There are many cases where we have seen long-term responders. I know you're thinking it is something will, but we have seen long-term responders. They are less in number, much less.
Look at
than in
Montessa.
other.
Yeah, Montessa. She gave me hope when I met her on Twitter. I'm like, no, because to me it was impossible,
Yeah.
But even my oncologist that had retired last year called me to tell me, you did it.
I see how grateful you are. I think that's
Oh.
what it is. Just count your blessings, live your life, and
Blessed
really
beyond
live-
measure. He
Yeah.
said that my immune system was very, very strong.
That's a blessing.
That's a blessing, because that's where we have seen long-term responders. Because patients who do respond to immunotherapy do it for a very, as I said, I showed you the curve, and 3 years, 4 years, they're
Right.
responding long-term. And
Right.
if your doctor is saying you're cancer-free, just count your blessings, show
Yeah.
gratitude, live your life. I really hope and wish the best for you. I think
Thank
you will
you.
be.
Thank you so much.
I told you it was a miracle. So to be
She need not.
2019, it wasn't until 2019 until Tecentriq came out. And when 2006, when I got sick, it was the standard of care for the past 30 years. Nothing, the needle had not moved. So,
And you were so young, and
Yeah.
you're still young, but I
I'm
mean, I
old.
remember, I know your story, I got your book, but this is like unbelievable. Yeah, that gave me hope. But then very little people, like maybe 2 people.
But we're getting there. We're getting there, Mary.
I know.
Oh, I'm
very
sorry.
happy.
Oh, look.
I've just finished 6 rounds of chemo, and it was kind of a mixed result in that the original tumors have pretty much resolved, but I have a bunch of small nodules that are new and a new lymph node. Am I someone who should be looking for a clinical trial, or should I let the immunotherapy that they've currently got me on ride it out and see what happens?
So you will most likely get a immunotherapy maintenance, and then upon relapse, if it does happen, then you will get which is injected in. But at that point, I think you can have this conversation with your oncologist about what clinical trials are available in your area and what you qualify for, because these are the 2 questions: what is available for you and what you qualify for. And
So are we talking about waiting for relapse or prior to that because the response was not so great?
right. So there will be trials available for relapse small cell. They're mostly for relapse. So you'll have to wait to not stop responding to your frontline treatment.
That makes sense to me.
See if there is any maintenance trial. There are not too many, but there are some that are in the maintenance regimen. I don't know if they are open in all centers. They are usually very investigator-initiated, boutique, smaller trials. So it may not be available for everyone and for every center, but I would write out the immunotherapy. I mean, because if you do respond to immunotherapy, it can be a long-term response, because people who do respond to immunotherapy can be a long-term responder, right? So you definitely should get that. And then for second line, the doctor will most likely put you on lurbinectedin. But at that point, it is a fair game to talk to your doctor about, what next? Do I have any clinical trials in this space where I could be eligible for and that you have open in this area? I keep mentioning, going back, since biomarker in small cell is not that well researched and well known, these decisions about clinical trials are very gray area. So, it is not black or white like we have for non-small cell. So you have to talk to your oncologist about what is available and what you qualify for, but it was most likely be in the relapse setting.
You'll be happy to know that I am participating in they do a blood draw every 6 weeks, where they're actually sending it to look for biomarkers. If they don't have
that's wonderful. I'm so thankful that you're actually participating in that. I mean, it's thank you so much. Thank you.
It's only a little bit of blood.
A little bit of blood's going to make a big, even tissues.
Big difference.
Let's go.
I was diagnosed over 4.5 years ago because of severe coughing, but never really had a lot of pain, well, much pain at all. I went through COVID and pneumonia this past summer and got through those really well, I think because of all the vaccines. The one thing I had it's been a year now, is 4 new growths in my vertebrae. I think I talked about this once before. Mm-hmm. And I was given Dr. Sen's stereotactic radiation for those. When I was diagnosed, it was a year before immunotherapy was even approved for small cell lung cancer. So I never received any of that. And like I say, the only treatment I've had for my spine, my vertebrae, was the radiation. I still get that was horrible pain. I couldn't believe how painful those were. I get twinges, but nothing more. But I guess my question is, should I also be waiting until, because I've been in remission now for 4.5 years, well, really almost 5. If immunotherapy for me then would not be proper unless my primary tumor starts growing again.
Yeah, well, these are really specific questions. And I think, it should be spoken with your oncologist who knows your case, definitely, because I don't want to make any clinical decisions in this platform. But I think most likely they will say, if you're in remission, then you should, only think about the next step once you're no longer in remission. Okay. They're not going to put more drugs in you if you don't need it, because you have to understand every drug has a side effect. So if you don't need a drug, why would they even give you a drug? It's not right. Good for, even the best drugs have come with their own share of side effects. So if you are in remission, then the doctor will say, the next steps need to be decided based on how your disease is responding.